The talks aren't the same, though.
That's what I've gathered. That's
Okay.
why I forced myself to come to the talk, so I can go.
Okay. This one will be more basic science. So does that work for you?
More basic?
Yeah.
Oh, good. Perfect.
Okay.
Good afternoon, everyone. I'd like to start this week's research conference. Amy, it sounds like my mic's not on. I'll just speak up. There we go. Well, first of all, I want to thank Swati Shah for suggesting that we invite Dr. Snyder while he's visiting Duke. Many of you may have a chance to meet with him, and if not, he'll also be presenting some of his work at Cardiology Grand Rounds later today. So I encourage those who can attend that as well to attend. It's a real pleasure to introduce Dr. Snyder, and normally I don't like to read a full bio, but many of us not in genetics may not be familiar with his substantial contribution, so I'll embarrass him and read this because it's really a tremendous body of work, and we're really happy to have him here today. Dr. Snyder is the Stanford Asherman Professor, Chair of Genetics, and Director of the Center of Genomics and Personalized Medicine at Stanford University. He received his PhD from California Institute of Technology and then postdoctoral training at Stanford. He's a leader in the field of functional genomics and proteomics, and one of the major participants of the ENCODE project. His laboratory study was the first to perform a large-scale functional genomics project in any organism, and has launched many technologies in genomics and proteomics. These include the development of proteome chips, high-resolution tiling arrays for the entire human genome, methods for mapping, global mapping of transcription factor binding sites, pair-to-end sequencing for mapping of structural variation in eukaryotes, de novo genome sequencing of genomes using high-throughput technologies, and many of these technologies have been used for characterizing genomes, proteomes, and regulatory networks. Seminal findings from the Snyder lab include the discovery that much more of the human genome is transcribed and contains regulatory information than was previously appreciated, and a high diversity of transcription factor binding occurs both between and within species. He also has combined different state-of-the-art omics technologies to perform the first longitudinal, detailed, integrative personalized omics platform of person, and use this to assess disease risk and monitor disease states for personalized medicine. He's a co-founder of several biotechnology companies, including Protometrics, Affamix, Excelix, and Personalis, and he presently serves on the board of a number of companies. Today, he will present an analysis of human genomes, transcriptomes, and regulatory networks. Please join me in welcoming Dr. Snyder.
Okay, well, thanks very much for having me here. This talk will be much more on basic science and how is it that we're different from one another. So when you look at each other or your neighbor sitting next to you, why is it that you don't look the same as them? And what are some of the principles behind that? Tonight, I'll talk a little bit more about personal omics profile, which will be both a personal experience and directions we're trying to go for trying to bring omics technologies into medicine. So I've shifted the title a little bit. It's still overlapping with the one I gave earlier, but I think we'll focus much more on the variation issue. And so the fundamental problem that, at least one of the problems that our lab studies is what's the difference between. Is that showing up? Can you see that or don't bother? No? Yeah, don't bother. Let's try this one. All right, that one you can see. Look at that, you get in stereo. Okay, great. So the fundamental question is, what's the difference between people and what's the difference between related organisms such as humans and chimpanzees? Now we know from the DNA sequence that people are about 0.1% different from one another, so one polymorphism every 1,200 base pair at the single nucleotide level. At the larger structural variation, so you may know that there are large deletions, insertions, and inversions that occur throughout the human genome. That's actually a much greater source of variation between people, and many of us like to think that this is probably responsible for much of the diversity between individuals. And that's something that our lab and other labs have worked on. Likewise, for the chimp, the difference between the chimps and humans, at least at the alignable sequence, is rather small. It's about less than 2%, probably about 1.6%, although there are some structural variation differences that lead to differences between these organisms. And certainly as people worked on this over the years, traditionally people have focused on the coding regions, thinking that differences in amino acids are primarily what's responsible for the differences between us or between us and our closest relatives, the chimpanzees and other organisms. But more recently, it's been, well, it was proposed a number of years ago by Mary Clara King that, in fact, regulatory differences may be responsible for differences between individuals. Rather, in fact, perhaps more so than amino acids. Now, at the end of the day, it's obviously going to be some combination of both, but I think many of us, including our lab, certainly like the idea it's probably more due to regulatory differences, and that's some of the work that I'm going to show you today. So there's been a number of studies to actually investigate this. It started initially, there was some individual gene analyses, and then later people started doing microarray analyses to study differences in gene expression between different individuals. And so there's some seminal studies mostly from Dermazakis and his co-workers that have shown differences initially with gene expression microarrays and then later with RNA sequencing. Again, showing there are thousands of genes that are different quantitatively between individuals. And this has also been shown at the level of species, that its difference amongst primates is quite extensive at the gene expression level. And so, again, we can see these differences, and then the question appears, what are these differences due to? And there are a variety of different mechanisms by which these differences in gene expression can arise. They could arise by differences in regulatory sequences, transcription factor binding. They can be due to epigenetic differences, and they can be due to post-transcriptional issues as well. And so what I'm going to tell you about is some of our work to get at all of these different issues, particularly the first two. And so what I'm going to tell you about is our work to actually try and understand variation in regulatory information amongst different individuals and also between humans and other mammals, including humans and mice, and how good are mice as a model organism for humans is a very interesting topic these days. I'm also going to say we see a lot we do a lot of analyses for trying to understand differences at the level of RNA and transcription factor binding and things like that, but how much of this is translated into differences in protein level, and that remains to be seen. And then at the end, I want to tell you about some new technologies we've been trying to implement to try and decipher accurate transcriptomes at a level nobody's been doing before. Typically, you use RNA-seq to look at parts of RNA, and what we're trying to do is look at complete transcriptomes. So to jump into this first topic here, which is trying to understand variation in regulatory information, this work, of all things, started from some experiments we were doing in yeast, where this was in about the mid-2000s, where people were sequencing lots of different genomes. This was becoming the thing to do, was to sequence lots of species of flies or lots of species of yeast. This is still while Sanger sequencing was prevalent. And people are sequencing these organisms and then trying to find the conserved regions between them and say, aha, here genes are pretty conserved between related species. But they would look outside the genes to find these other elements and say, aha, here are the conserved regions. But, and these must be the important parts. These must be the regulatory sequences. And I think the prevailing wisdom at that point was that regulatory sequences are conserved between related organisms, and this is how you find those. Well, our lab was one of the ones to invent a procedure called chip-chip, which is doing chromatin IP, where you pull down a factor with its associated DNA and you probe microarrays. And then more recently, this was morphed into chip-seq, where you do the same thing. You pull down transcription factors with their targets, and then you sequence and then map back those reads of the genome to map binding sites. And what we discovered at the time, which was a surprise, was that very closely related organisms, in this case, these are closely related yeast, Cerevisiae, Mycetae, these are about the same distance as chimps and humans, thought to be about 5 million years apart. Bayanus and Cerevisiae is thought to be about the same distance as rhesus and humans, roughly to give you that scale. It turns out most of the binding sites for these factors that we were studying actually did not overlap, only about 39% between any two organisms. So in fact, binding sites were not conserved. And in hindsight, that makes quite a bit of sense because we don't look exactly like a mouse. and it makes sense that we shouldn't be identical. So we then took this, and by the way, if you looked at all 3 species, they would be about 20% or so identical between the 3 individual species. So the point is there is extensive divergence in regulatory information between species. We then took this information and then started looking at humans and looking at variation within a species. And we did this for both yeast and humans. Yeast where you can use great genetics, and humans we care a lot more about probably than yeast. And so we wound up studying both of these. And we started using these lymphoblastoid lines. They're called LCL lines. Many of you are I don't know is this getting too technical? Does everyone know what a lymphoblastoid line is? Okay. How many don't know what a lymphoblastoid line is? Okay, some. So these are actually like pre-B cells that you can immortalize with EBV. And they use a lot of basic research because you can take someone's blood and you can immortalize these lines. And then you can analyze them between different individuals. So you can think of them as pre-B cells. And what's special about them is that they're now getting sequenced. So there are thousands of these lines that have been sequenced from different individuals around the world. So they're a really terrific resource because they have all these sequenced genomes, and you can relate genetic information to molecular phenotypes. And so what was done here is that we actually mapped the binding sites of this factor, NF-kappa B. So that one you've all heard of, right? Who has not heard of NF-kappa B? One person. Okay, a few. All right. NF-kappa B is probably one of the world's most famous transcription factors. Sorry, I'm trying to calibrate myself, and then I'll pitch accordingly. So probably nearly every scientist in the world works on NF-kappa B, and they probably might even be working on it and not realize it because it controls apoptosis, it controls inflammation, it controls every single important biological response you can think of. Anyway, it's a transcription factor, and it binds 15,000 sites throughout the human genome. And you can map the binding sites of this by pulling down this factor with an antibody. And then actually what you do is you pull this down along with its associated DNA, its target DNAs, and you sequence and you map them back and you get these peaks. And what we discovered is that actually there are quite a few variable binding sites amongst individual in these cell types. So these are all similar types of blood cells, but they all have very different, but there's a lot of differences in binding sites. In fact, about 7.5% of these sites will vary between any 2 individuals at the statistical thresholds we've used. Okay, so what this says is that there's quite a bit of variation in difference in binding of this factor, which presumably leads to differences in people's response to stimuli of NF-kappa B, and we can actually show that in other ways. We also looked at a general factor, polymerase II, which is the main enzyme. How about that one? Does everyone know that one? Yes, okay, good. So this factor obviously controls gene expression in genome. And the bottom line here is that there's a lot of variability in binding sites. And what was kind of interesting about all of this is that, in fact, if you looked at the binding sites of these factors, they were very, very interesting loci. So, in fact, the binding site I'm showing here is at a gene involved in schizophrenia where there's incredible heterogeneity here, even though there's a blood cell. And this one actually shows up at an immune response gene. A lot of these variable sites are genes involved in functions that would be of interest. So the next thing you can do then is you can actually take this information and you can try and relate it to the genetic information. These cell lines have been sequenced, so we can try and see if you see a difference in binding, how does that relate to a difference in the sequence that underlies it, and specifically in the motif that NF-kappa B binds to. And so what we can do, this is the NF-kappa B binding motif. I see things are a little washed out here. But you can start correlating in these peak regions differences in the sequence here with differences in these motifs. And what you'll do then is you can actually see, so for example, if you have a G, which is a consensus site for the binding site for this factor, you will in fact get a nice binding signal. And if you switch one to an A, it goes down. And if you have 2 A's, it goes to 0 at this particular locus. Okay, and so you can start correlating this across hundreds of sites. Okay, and same for Pol2. In this case, because Pol2 is a general factor, you actually look for some of its associated sequence-specific factors that help it bind. And we did find a difference in some of these, not as many. Okay, by the way, here's another NF-kappa binding site. So again, if you start changing the consensus sequence, you will lose binding. What was interesting, though, was actually only a subset of these factors, or these variable binding sites, will have differences in the binding motif for this factor. And the reason for that is that transcription factors work as a community. A group of factors work together to actually bind loci to be able to turn genes on and off. And this is one of the complexities about our human genome circuitry. That is to say, different factors come together to regulate different genes. And people are trying to decipher this. This is called the so-called regulatory code. And so there are projects out there like the ENCODE project. How many of you have heard of the ENCODE project? Okay, it's about half. This is a project where they're actually systematically mapping regulatory information by either open chromatin or mapping the binding sites of all these regulators. We're actually part of this. So we're mapping the binding sites literally of thousands of factors. There are 1,400 in humans. So I guess it's 1,400 is the goal across a number of different cell types. And the ultimate goal is to figure out who works together with who to regulate what. And at these variable binding sites, what we discovered is that a few percent of them actually have differences in the motifs of the factor itself, but many more are actually in partners of the factor. That is to say, here's one example here. So you may not have a difference in the binding site for this factor, but you could have a difference in the binding site for its partner, and that would affect the binding of the original factor. Okay, so if your partner doesn't bind, you don't bind, is the idea. And we could set up bioinformatics tests for this to actually discover partners for factors and where they worked. And what you do is you set up a so-called co-association test where you find the variable sites for this factor. They don't change in the binding site for that factor itself, but you look for motifs that are associated with this factor but don't belong to that factor, and their variation correlates with binding here. So it's a so-called covariance test. Okay, I don't know if this is getting too jargony or not, but I'll try and show you one example of this. This is a factor, well, by the way, at the end of the day, we discovered a lot of new partners for NF-kappa B of who works together with who. And basically, this shows one example. This is a case where, with this factor, this is a partner of NF-kappa B. This is NF-kappa B binding. And the NF-kappa B binding site itself doesn't change, but its partner site changes. And you can see if you change, in this case, 2 changes, where you change that C and that G, each of them to an A, you completely abolish binding in these different individuals. And so the net result is that you can go through and you can find partners like this where there's an associated motif, and when you change them, you lose binding. And we can show that that factor really does bind there. And you can then correlate this out. So the net result is you can find the various binding sites through the factors themselves or their partners to see where you're seeing variation and who's regulating who. Now, why is this important? Well, ultimately, we want to correlate this with disease states. So here is, and so we've done this systematically. We can take, there's a database, and I'll talk more about this later at 5, that a tool Butte's lab has put together called VeriMed, where they've in fact brought together, they've been reading the literature for now about 8 years with a team of curators, and they're reading all the GWAS studies, and they're calculating likelihood ratios. And basically what they've been doing is putting these into a database, and you can actually correlate the risk alleles in their database with the binding information I just mentioned. And so this is a case where here's a risk allele for atherosclerosis, so that should interest this group here. This is the sequence, this is the NF-kappa B binding site, and then you can see if you match the consensus, you will bind. That's actually increased risk. It's actually probably causing inflammation. And here's decreased no binding, which is associated with the A, the decreased risk allele. And so at the end, you can see people's risk allele in regulatory information associated with this. Why is this important? Well, when you get your genome sequence, how many of you have had your genome sequenced? Only one so far. How many want to get their genome sequenced? Okay, if you want to learn what you might learn from a genome sequence, you can come to the 5 o'clock talk. But the bottom line is, if you get your genome sequence, you will want to incorporate this kind of information to be able to better understand your risk allele for certain diseases. And so that's one of the directions that we're taking this. Now, another group, Jonathan Pritchard, working with U of Gilead, has actually extended this work further. They've mapped the binding sites for open chromatin, which are actually regions where regulatory sequences reside in the human genome. And they've done this for 60 or so of these lymphoblastoid lines I mentioned, these called LCL lines. And they can actually map, again, these sites, and they can determine precisely where some of the regulatory sites reside. And so here's an example of, this is one of these open chromatin regions. It shows up in individuals that have this sequence here. They have A's at this site. Actually, it's probably hard to see, but you get a slightly decreased signal if you change one of those to a G. And if both are a G, you lose the signal completely. You can actually zoom in and see even what bases, where this is binding based on the maps here. And the bottom line is they could actually map about 9,000 of these regulatory sequences throughout the human genome. So you can take that information again, and you can map it onto personal genome sequences and try and understand which allele you are relative to what disease risk you might have. So that was a very nice study, but for us it left many open questions that we felt weren't resolved. So as you may know, there's lots of regulatory elements throughout the human genomes. There are enhancers, there are promoters. And one question is just how variable are these amongst different people? This says in general regulatory sequences vary, the studies I told you. But the question you may ask is, what kinds of elements are likely to be the most variable? Is it the repressed regions? Is it the enhancers? Is it the promoters? And what happens when you switch? Can enhancers switch to promoters and then promoters switch to enhancers? How does this regulatory information change when it does vary between people? Okay. Another question is just how does changes in regulatory sequence map to changes in protein levels? And these are some of the questions that we've been getting at. And so one way to get at the first questions was, in fact, to map chromatin marks, which are nice signatures for enhancers and promoters and repressed regions. You can do this throughout the human genome using this chromatin immunoprecipitation method I mentioned earlier. And specifically what we did was we mapped all the marks. These are chromatin marks, if you will, modifications that are either at promoters. So ones in boxes are things we mapped. So we mapped these in promoters. We mapped a mark for gene body. We mapped several for promoters, actually. We mapped 2 for enhancers. One's for active enhancers and the other's for all enhancers. We mapped other interesting elements in the genome. They're called CTF sites. They're actually boundaries that set up domains in the nucleus. Cohesin, which is another interesting molecule that works with CTCF. We mapped marks for repressed regions as well. And so we mapped these in 19 different individuals. These are some of the sequence lines I mentioned before that come from this, it's called the 1,000 Genomes Project. So we mapped them from diverse populations, so Europeans, Yorubans, which are a fairly diverse population, Asians, and then we actually had access to a number of individuals from Bushmen in Africa, which are a very divergent group in the human population. And we went in and we actually sequenced their genomes first for these 4 Bushmen. It's actually an incredibly accurate genome sequence we put together for these. So at the end, we can map all these marks. And we varied a little bit with the individuals, somewhere between 9 and 13 different marks of either chromatin marks or transcription factors in these 19 lines to see what kinds of diversity might exist. This next slide just shows examples of what you find. So there are cases like this one where the mark is quite constant. And here's another example where the mark is very similar between different individuals. This is an enhancer mark. But you can see there's actually a region that's highly variable here where the mark is showing up. This is a population-specific mark. It actually tends to show up in Europeans and also to some extent in Asians, but not so much in either the Africans or the Bushmen. Okay, and what was interesting is actually that 33, this is a mark for enhancers, at the threshold we used, about 1/3 of your enhancers actually vary amongst individuals and 2 or more individuals in the population. So there's quite a bit of variation of enhancers or regulatory sequences throughout the population. Now the next part, it could get a little esoteric, but it's actually kind of interesting from a bioinformatics standpoint. You want to be able to look at this very systematically. And so what you can do is you can divide up the human genome into segments, different regions. I gave you the names: enhancers, promoters, repressed regions, and CTCF sites are the names, but you actually divide them into active enhancers or weak enhancers or poised enhancers, where you actually, these are thought to be enhancers that are ready to go, but they actually have both a repressed mark and an active mark. They're conflicted, if you will, enhancers. And they basically have these different marks. And so you can take the regulatory information we have and bioinformatically segment, it's called, the genome into compartments. So you can divide these up based on the types of marks they have. And so as an example, here's a transcribed region in the genome. This is transcribed weakly. That'll be the case if it only has this particular mark. It's called H3K36 trimethylation. This is primarily a promoter mark. This one here, a very strong promoter, although it can show up at enhancers too, this trimethylation mark, and so on and so forth. So you can actually see, and you can informatically, and you can use machine learning tools to be able to divide up the human genome into its specific segments. Okay, so imagine now you've taken the genome, you've divided it into segments, and then you can ask, how did those segments vary amongst one another, amongst individuals? And this is how we do this here. So this probably is a little complicated, but each of these segments is, again, a different mark. So the red ones in orange are promoters, the green are transcribed regions, enhancers are the yellow ones, and so on. And so here's a gene that's actually transcribed from right to left. The promoter is in red, and it's pretty constant. The enhancers are actually in yellow, and they're in the first intron for this, and actually in the 5' end. And they show up there in the gene body, and that's all pretty constant, right? You look at a gene like this, you can actually, this one I believe is transcribed, let's see, it must be, I thought it was left to right, but now I can't tell from this mark. I guess it's actually right to left. But there is a variable promoter here. The transcription itself, the transcribed region, is very variable amongst these individuals in the same kinds of cells. And you can see there's just a lot of variability, both the regulatory and the transcribed regions as well. So you can say these different regions, either promoters or enhancers or what have you, are varying. And at the end of the day, you can tally this up and you can ask, what are the most variable regions in the human genome? Are they promoters? Are they enhancers? Are they repressed states? And the answer is, actually it turns out active enhancers are by far the most variable parts in the human genome. Okay? They show a lot of differences between people. Again, and at least it's more than a third. It depends what you use the threshold. It can be up to 50% or more. Okay, let's see. The active transcribed regions are probably less variable, but the enhancers are quite a bit variable, and the poised enhancers are and transcription start sites are actually very variable as well. So you can find again that the enhancers are highly variable, promoters less so, and others less so. Repressed regions are actually the least variable regions in the genome. So when regions are shut off, they're generally shut off amongst most people. So the next question is, do enhancers and promoters actually interchange between one another? I actually thought they would a lot. Why did I think that? They have a lot of marks in common. They have a lot of activities in common, like transcription itself. And much to my surprise, actually, you see very little of that. Enhancers rarely convert to promoters and vice versa. There's a very complicated way of showing this, but it's the simplest way I can show it. So bear with me. These are the different states. Again, this one would be the, let's see, this is the transcription start site, okay, active and less active. And so if enhancers were converting to active regions, you would actually see a strong brown signal here, which you do not. But instead, you'll see that what these are just the regions that are changing are shown up here. Instead, active enhancers will show up as repressed regions, actually. And vice versa, or similarly, I should say, promoters also will switch. They don't switch to enhancers. They also will show more often than not as repressed regions. You can actually see conversions between weak and active enhancers as well. So what's happening is, as you get to differences between people, the biggest differences aren't in them changing the types of elements they are, but rather the activities of those elements. So enhancers will convert to promoters and vice versa. Now, why is this important? Well, a lot of this has functional, well, before I get that, it will have functional consequence. I'll show you that on another slide. Okay. This is saying that actually what happens in parents often happens in kids. If you look at regions marks in kids, most of those marks are inheritable. I plotted them all on here. Again, it's a complicated slide, but you can see there's generally correlation between children and their parents. Well, basically that's what this shows. It's probably easy to look at correlation coefficients. In general, these different marks will be highly correlative between kids and their parents. Okay, so what that means is that you're actually inheriting these chromatin states across individuals. Now, that could be either epigenetically or it could be due to genetic changes. And we think a lot of it's genetic changes. So the genetics is actually driving a lot of the epigenetics, we believe. And the reason for that is we can correlate with SNPs, again, these polymorphisms in these regions. And we can take this one step further. We can correlate it with specific SNPs. And this shows for all these transcription factors. So the ENCODE project has mapped lots and lots of these transcription factors in these cell lines, at least in one of the cell lines. You can see which ones are bound by which factors and which ones for which they're motifs. And all of these regions here, so all of these EBF sites, each of these is a different site. Each column is a different site. These all have a variable histone mark that lands smack in that motif. So what that's saying is that you can get variation right in these marks, and that will lead to a variable chromatin state. Why am I telling you this? Again, if you have one of these sites, you will probably correlate with a certain chromatin state, and that will correlate with activity of a particular gene. And so we're trying to bring this kind of information in the personal genome annotation, and again, I'll show you more of that in this afternoon's lecture for those of you who go. Now, some people say, Mike, that's great. You see variation in chromatin states. Who cares? Most of these sites probably don't do anything anyway. And we would argue, well, sometimes that's true, but sometimes not. That is to say, here's a variable histone enhancer mark. And you can see it correlates pretty nicely with gene expression measured by RNA-seq. So here's the same gene, okay, right here. I show you these are actually a series of enhancers because this is a very small scale. There's actually at least 3 enhancers in this region. And you can see variability in activity of these enhancers correlates with the amount of RNA that comes out. It's not a perfect correlation, but it's pretty good. Okay, and in general, we can plot this overall. That is to say, there's a pretty good correlation between expression and these activity marks that you see. Okay, again, it's not perfect, and it turns out overall, if you only have one variable enhancer, you usually don't see a difference in RNA, believe it or not. You actually need multiple ones, and we can show that correlation too. So the net result is that we actually have a lot of variability in our regulatory sequences. When you get multiple copies of this variation, you'll actually lead to differences in gene expression. And again, we think we can correlate this in interesting ways. One of those ways is if we look at variation in chromatin variation, it does correlate with differences in gene expression. We can show this another way. These are expression QTLs that are related to expression amounts. So we can show there's a correlation between all of these variable regulatory marks, if you will, and differences in gene expression. It's related to what I was just telling you before. We can show it relates to differences in the DNase hypersensitive sites I mentioned before. And perhaps most importantly, what would interest groups here is that it actually also correlates very strongly with disease risk loci. So you can start correlating regulatory information with disease risk loci. Now we have other studies from ENCODE data where we can map how much of the GWAS hits. You may know that something like 88% of GWAS hits do not lie inside genes, actually lie outside the genes. And it turns out a substantial fraction of that overlaps, depends how you define it, overlaps with regulatory sequence is defined by ENCODE, and we can also show by these variable chromatin states. At least we can actually correlate at least 30% of it with differences in that. This gets a little complicated. It doesn't prove they're causative SNPs. They're simply linked. Okay. So is everyone able to follow that so far? I don't know if this is getting too heavy-duty basic science or if this is too, but we're very interested in, again, this because ultimately we want to correlate disease with genome sequences with regulatory information, an area that's drastically lacking. All right, so we know there's differences in gene expression. We know there's differences in binding and chromatin states. How does this relate to differences in protein levels? So here what we can do, no one's actually really looked at this before, and a postdoc in the lab, Linfeng Wu, actually set out to take this on in what was really a rather Herculean effort here. She basically mapped protein levels, relative protein levels, in 90 different lymphoblastoid lines. Now the way you do this is by something called TMT labeling. You can put mass spec tags on your whole collection of proteins. So you take the cell lines, you make protein lysates, and you cut them into peptides, and you ask, and you label up all the peptides, well, all the ones with, well, they all have amino groups, with the TMT label. Okay, this label actually adds a mass tag, and the mass tag will basically be one different for each of the different samples. Okay, and it's done in such a way that originally the mass tag is the same mass for all of them, but when it fragments, it shifts by one. And this gets a little technical, but the bottom line is at the end of the day, you can mix all these things together. They migrate perfectly in the first dimension of mass spectrometry, but in the second dimension, you can quantify the differences between the different amounts of protein in a relative fashion. So all you really have to remember is that normally you can follow about, we can follow about 5,000 or so proteins in these mixtures. And we look at how much their relative amounts differ, and you can do it with these different mass tags. Very cute trick, actually. And it was invented by Rudy Ebersol's lab, the original concept, although the version we use is done by a company, Thermo Finnegan. So you mix these together, and you do this mass spectrometry, where you can actually see differences in protein levels. And the trick is you actually have to mass spec as it's not as good as RNA-seq. You actually have to run the experiments 3 times. And we actually had to do 2 to 6 biological replicates to get nice, robust data. It's a lot of work. At the end of the day, we were following about 6,000 proteins across these 90 lines. Again, this is a huge amount of effort. As a side note, the machines you use for doing this, they cost about $1 million, and they become obsolete in about 2.5 years, and so we now have to buy another $1 million set of machines that work better. So I've learned my lesson. We're going to be renting them for the future. Okay, and although that's not cheap either, but it's still cheaper than having $1 million doorstop. Okay, so anyway, we followed this many proteins, and even though the threshold we used here was 1.5-fold or more, only 6% had varied. Now, if this were RNA, that number would be something like 25% or so. So the amount of variation at a protein level is actually much less, we would argue. There are caveats with what I just said, but in general, we think the amount of variation that occurs at a protein level is less than an RNA level. Why is that? Because we think there's buffering that occurs, that you actually will compensate and adjust changes to make it for a more even amount of protein level, but there's still substantial differences. Now, the reason we did all these lines is so that we could, in fact, map genetic changes associated with protein levels. It's a GWAS type study. I'll show you the results, but if you need more details, you can talk to the experts here. Swati is one of the pros, so you can get some lessons from her. But the bottom line is we actually map then differences in protein levels, they're called pqtls, protein quantitative traits, across the whole genome. And it depends obviously on the threshold you use. And at a 10% false discovery rate, we got 77. It's not nearly as many. There would be thousands if this were a gene expression experiment. 273 with a higher false discovery rate. And that's because these experiments are noisier. They're not as robust as the RNA-seq experiments. But nonetheless, we can map quite a few. the net result is we can then say of the differences in protein levels we see, how many of them show up as the same locus, the same spot that are showing differences in RNA expression. As you might imagine, if you have a difference in RNA, like it could be all your differences in protein levels are due to differences in RNA, right? So it could, doesn't have to be. And in the end, about half of them overlapped. So half of our, when we see a difference in protein level that we can map genetically, half of them map to differences in gene expression level in the same cells. But the other half don't. That means they're being controlled by something other than RNA levels, okay? And so and just to show you one of these, this is a very interesting gene that's involved in, now I've forgotten. Well, nice thing is you can Google it up yourself right now if you want. And the bottom line is I know it's something medically interesting, and I've forgotten now. It's embarrassing. Okay, but the bottom line is actually this one, the highest SNPs for this actually map near the 3' end of the gene. They're not anywhere near the regulatory elements for this gene. And in fact, here's the original locus here. And when we zoom in on this, we actually think it's right in the 3' UTL, in fact. And then there are others we can map the differences in splicing sequences and such. So we can actually map quite a few of these, well, half of them outside of the gene expression differences. Why is this important? Well, these are genetic changes, again, that we can correlate with differences in protein levels, which, again, is very useful for annotating human genomes. Because at the end, if you have the allele that matches with high protein level, you want to know that versus low protein level, and we can correlate that with medical outcomes. The other thing we can do with all these data is we can start looking at this at a more systems level approach. And one of the things we've done is we can take all of the variable proteins and we say which ones correlate the most. If this protein's variable, what other proteins vary like this across these 90 cell lines? And it's a somewhat simple correlation analysis, although it requires a lot of controls. And the net result is we can find 5 groups of protein that actually show a lot of co-correlation. So it turns out a lot of metabolic things will co-correlate, and what that means is that they're actually showing similar differences across individuals. So it turns out differences in glycolysis at the level of variation in glycolytic enzymes are differing across different people, and the whole suite will differ. Same is true for translation, ribosomes, other translational processes, and a lot of complex. Here's one involved in DNA replication. So you'll see groups of proteins, if you will, that will vary in bulk across different individuals. And we can tease that out from these sorts of studies. You can also correlate that with differences in RNA, and there's some correlation between the variation in protein with the variation in RNA, but most of it is not. It's very different. There are a lot of differences between proteins and RNA, and I'll be mentioning some of that later today. The other thing we can get at is what's the differences between species. I've been talking a lot about within humans and within a species. What about between species? There's some very interesting papers that came out last year where people were looking at differences in gene expression between mostly differences in primates, but also other outside primates as well, mice and other organisms as well. And the conclusions from those studies, this is just one here. This is from Chris Berg and Blenko's lab. They found that in general, gene expression is similar between tissues of different species. So in general, the expression profiles would be of a mouse heart would look very much like a human or a chimp heart, rather than similarities between tissues, between species, sorry. But they found lots of differences between splicing amongst the different species. So they say gene expression is similar between tissues. Now, as part of this ENCODE project I mentioned, where they're mapping lots of transcripts and regulatory information in different cell lines, there's another project out there called Mouse ENCODE, which you may or may not have heard about. That's mapping RNA across lots of different mouse tissues. It's also mapping transcription factor binding sites and things. And we've shown, just like a lot of what I mentioned here, a lot of regulatory information varies between mouse and human. But the surprise was actually when we looked at RNA differences, we mapped the RNA expression across many different tissues. In fact, 14 different tissues were analyzed in both mouse and humans. And what you're supposed to recognize here is that human are circles, mouse are these pointy triangles, these hats, if you will. And the colors are the different tissues. And what you'll notice is that the tissues aren't clustering together. The species are clustering together. What does that mean? That means actually a mouse intestine is more similar to a mouse heart than it is to a human intestine. So this is very different from what other people have published, and it's kind of, so naturally it wasn't very well received. So because it's saying that organisms are more similar, tissues within them are more similar to each other than, again, between species. And if you think about mouse as a model for humans, it's not welcoming news. So we've looked at this further. First, it could all be technical artifacts done in different labs, even though you can see there's quite a few labs that were involved, at least 4 different labs, and they all do cluster the way I've said. So we went in and did it all ourselves, same lab, repeated all of this. This is after 10 tissues, and we've now added 3 more tissues. So 13 different tissues, same thing. The mouse ones all cluster together, and the humans, sorry, I think that one's human and that one's mouse, but you get the point. They're actually clustering by organism, not by tissues. So what's going on? So we spent a lot of time trying to figure this out, the difference between our results and other results. The other groups primarily looked at a few tissues. Ironically, some of them didn't look at humans, but they looked at mostly these 5 tissues here. Well, it turns out these 5 tissues are the most divergent ones out there. And in fact, we can recapitulate. So this is what others have primarily, you can't see that. So we've looked at how many unique genes there are in these tissues. And sure enough, again, most of the distinguishing genes, the 5 tissues they studied, are in fact the most different you could have grabbed to be able to do this. And then we can repeat our analysis with just using those 5 tissues. We actually will get, to some extent, what they find. That is, if you just use those 5 tissues, you can get things that cluster by tissue. That's because they're so super divergent. Okay? But if you do bring in, again, all of the tissues, you will see this. So then we said, well, there must be some level of tissue clustering in there, and in fact, there is. So we would say 75% of the thing that's driving these things together is because the same species. But if you look further, this gets complicated into digging down into the principal components further. This might, again, be more for the aficionados, but you actually can find clustering by tissues. It's just not the most dominant thing in there. You actually have to go down to what's called the 4th, 5th, or 6th principal components. It's in there, but it's at a very lower level. You can actually find the tissue prevalence. So this is mouse and human tissue. I can't remember which is which, but you get the idea. These different tissues will be associated with one another. Okay, so then the question is, what kinds of things are driving this? Why should a mouse be different from a human? What kinds of genes are driving that difference? And so you can ask, well, what kinds of things are most correlating in the mouse or most correlating in a human, but are different from the other species? Okay, and so you can tease that out informatically, and what we can find is roughly about 2,700 genes that are more highly expressed in humans over mouse, and vice versa, about 2,400 in mouse over human. And we can ask what biological processes these are in, and so these are things that are common across the different tissues, but definitely more in one organism than the other, and the answer is it's mostly metabolic genes. Okay, so at the end, you'll say, of course, this is obvious. The metabolism of a mouse is different from the metabolism of a human, and that's why they're clustering the way they do. And that's why the tissues of a mouse are more similar to a mouse than that of a human. It's also some other processes as well. But why do I like to show this? Well, first time I've actually talked about it, so you can shoot me later if you want. But I think the reason this is important is that the metabolism of a mouse is different from the metabolism of a human. And at the end of the day, they are different organisms. And yes, these are good model organisms for one another, but don't forget that they're also quite different. And that a mouse heart is more similar to a mouse intestine than a human heart is a human intestine. because its metabolism is very different. Again, that's going to disappoint a lot of you, perhaps. How many of you are mouse modelers? Use mouse models. Oh, you guys, only one person? Yeah, you guys drink the same Kool-Aid, then you'll be fine. All right. So anyway, I thought this was a very amusing story. And if you actually subtract out, by the way, all the species-specific genes, you can reconstruct the tissue-specific clustering again. So the point is, there's this bolus of genes that are very organism-specific that are driving organism-specific biochemistry, if you will, and differences. So I think that's kind of fun. Now, I see I didn't leave much time for the last part, but we've been thinking a lot. Our lab does do a lot of technology development, and one area that's always bothered me, we're actually one of the inventors of RNA-seq. There are several labs that have done this. And if you think about RNA sequencing, and I've presented a lot of this in this talk, what you do, for those of you who know, you actually take RNA, you fragment it, you then make cDNA copies, and you sequence that, and you map it back to the genome, and then you try and reconstruct the genes in the first place. And if you think about it, that's a pretty crazy way to do things, right? We'd be much better off. We do it because we can't get long reads. So we've started playing, this is my way of showing this. This is RNA, blowing it up into fragments, and then sequencing the fragments to get, and then you reconstruct them. So we've actually started using packbio to see what we can learn by generating long reads, and we do this in an amplification-free fashion. So we can get very long cDNAs, and we think, actually, there's not lots of time, but basically we took human DNA, we took 20 samples, or human RNA, we took 20 samples, we pulled it together, and we generated large numbers. The problem is the throughput isn't as high with this packbio, but you can still get reasonable numbers of very long reads. And at the end of the day, you get lots and lots of full-length transcripts. This is a gene that's reasonably long. I think it's about 4 kb or so in size. And you can see that for this gene, you get mostly large, not entirely full-length transcripts, but you get quite a few full-length transcripts with this technology. Why is this important? Well, if you look at a number of genes, you can actually find that 10% of the isoforms you find are novel. And you wouldn't find, it's hard to find this now with the short reads we use because you don't know which exons are linked to what other exons. You can get junction sequence, you know the neighboring ones that are linked, but you don't know within a full-length transcript which ones belong with which ones. And so this, by reconstructing the full-length transcript properly, or getting the full-length transcript properly, you can actually see. So we can say actually that this exon is in fact with this fuller length exon and not with the short one. We've done lots of reads, for example, with this. And you can see who always belongs to who, so you're getting accurate isoform description. Why is this important? Well, one of the things we're doing now, and I'll mention this in the later talk, we have a lot of allele-specific expression in us, where our mother's gene and our father's gene don't express the same. We need this kind of information to be able to correlate which splicing isoforms correlate with which genetic variations, to be able to see who's linked to who, so that we can ultimately try and reconstruct accurately molecular transcript profiles from individuals, and then ultimately correlate that with disease risk. So I'm going to just close by saying we can get very accurate RNA sequences now, we think, for 2.5 kb. There still are throughput issues. You always get complete transcripts at 3' ends, and they're mostly complete at the 5' end. Sometimes they're about 60 bases off when you get up to about 2 kb or so. But even you can actually get reads up to 7 kb. That's a median length now. Some complications with that. But you basically can get very long reads. And so I think in the future, we're not going to be generating these short RNA fragments. We'll be generating much longer ones to get much more accurate transcript profiling. All right. Conclusions, take-home lessons. First, there's extensive variation in regulatory information amongst humans. Enhancers are the most variable sequences out there. Protein variation is also extensive, but not as extensive as RNA. There is some buffering that goes on. Mouse and human tissues are more similar within a species than between species, I would argue. And lastly, we think that generating long transcriptomes will be very powerful. So this is the people who did the work. So Maya Kosowski led a lot of the, and her coworkers led a lot of the transcription factor and regulatory variation work. The protein variation was primarily done by Lin Fang Hu and Sophie Gandel, and very close collaboration with Hua Tang. The mouse human expression really counts on the whole mouse and ENCODE project. We also brought in some REM-C data from Joe Ecker. Tom Gingeris is an important part of this, but it's really spearheaded by the Lin brothers, if you will, Xin and Ying. That's true, they're really brothers. And the long transcriptome work was done by Hagen Tilder, Don Schrom, and Fabian Gilbert helped out. So happy to take questions if you have any.
Talk. I have a question. I was struck by what you said about the in combination differences that you see. And I was, we're all disappointed a little bit by GWAS and how you get very low signals, and part of it is because we don't have enough people, but we are just looking at one gene at a time. And I wondered before, and this reinforces, what if you could look at 2 genes at a time? Has anyone done the sort of power calculation to know how many samples you would need, and do you think you'd find more GWAS? I mean, it's always nature nurture.
Yeah, it's been hard to do that in a perfect pairwise fashion. So people haven't been doing that as much, to my knowledge. I think people are starting to do it. We're trying to do a little. Rather, what people have been doing now is the pathway analysis for GWAS. And so, and that's looking promising. I didn't talk about this because we're not quite ready, but we have a study with autism, actually, where we have taken, we look at burden, if you will, all the genes in certain disease and map them onto modules that are omically defined, if you will. They're interaction network modules. And so that's sort of a pathway. It's an unbiased pathway as opposed to, right now you curate the literature, right, and you get the NF-kappa B pathway or the TGF-beta pathway. That's a literature pathway. That's probably not a real pathway, by the way, but it's one we'll take as textbook gospel truth, but it's probably not real because the real one has lots of other components. So we reconstruct our own, and then you can map diseases on top of that, and that actually looks like it's a lot more promising. And so other groups are trying similar things for other diseases. As I say, we've been doing this for autism, but I think there are other groups as well. So that's the way to actually, we think, to get at this. And there are other tricks people do as well. So the complication is, and I'm sure anyone will tell you as a GWAS person, is that you lose it as soon as you go across the genome and you go all SNPs across the genome, you lose your power. You can't, the whole multiple hypothesis correcting part of it. And so what the tricks people do is they zoom in on promoters, proximal regions, because that reduces your space and then reduces the power you need, or increases your power because you're looking at a more narrow region, or you bring in the pathway. And I think both those, the first approach has helped us a little bit with some things, but the second one, I think the pathway is going to be the future. And I just think we have to redefine the way we do pathways. I think we're not doing it. We're still too textbooky. That's not the right way to take it, I would argue. Okay, I see others nodding their heads, so I must have hit a good note here. Yeah.
The binding thing, the variation in the binding sites. Have you looked at variation within individuals? In other words, if you look at that distribution, I'm not sure exactly what the distribution is, but if you look at it across at different time points, how much variation is?
Yeah, that's so right. Within an individual, have we looked at it? Yeah, we haven't, and that could be a caveat with these. That is to say, maybe these cells are frozen. They're not exactly the same cell state. But as far as people can tell, they certainly lump them together, that these are the same. So I guess I'm working under the assumption they're under the same cell, but that would be a caveat with our results. I think the fact, at least the ones that we can correlate with a genetic change, is a pretty strong argument that the difference is really due to an underlying genetic effect and not due to a timing effect. But I can't prove that, but I think that would be the most, for the ones that have a genetic change associated with it. And by the way, that's just a subset of the sites. It's not all the sites. So it could be the others are more of a timing issue, I suppose.
The other question is more of a technical kind of thing, which is when you're looking at that variation in proteins, and you said only 50% could be explained by, I guess, the gene
RNA.
expression.
Yeah.
Have you looked at RNA profiling ribosome profiling.
Yeah.
To see if that can actually explain or better elucidate. how
Yeah,
the
we just collected data on 90 lines, most of them the same lines I just mentioned for ribosome profiling. It's a great question. And the answer is I don't know yet. We're pulling the data together. We'll hopefully know very soon. But it's a great question. Yeah.
I would relate this to what the DCRI does, which is large clinical trials. We look at one compound and we test it in 50,000 people who are different ages, different races, and then we do proportional hazards and see if the drug works or not. So it's very binary. Do you think moving forward, like that seems completely different from what we're seeing in the basic science? Do you think that a single patient could in the future serve as like the basis for whether a drug works or not, or how do we do clinical trials in the future with?
Yeah, so I think they're going to marry up quite nicely. I would argue we're in the early days right now. What you want to do is take all of the data I presented here. Mind you, you want it in other tissues as well, not just in LCL lines. But we actually think that there's somewhere, well, as it currently exists, there's about 6,000 to 7,000 medically interesting genes in the human genome. There will be more as we learn more, but that's how many there are now. And we've actually, many of those are involved in pharmacogenetics and affect drug response. And so we can actually match a number of these regulatory variants now with pharmacogenetic variants. And we're still putting all of that together, but that's going to be a very, very powerful set, matching the variation in regulation on top of the variation in drug response. And I can tell you, if you don't learn anything else from getting your genome sequence, you will learn something about your drug response information. We're going to learn a lot more when we add the regulatory information on top of that. In fact, a lot of the well-known variants, one of the warfarin variants, for example, is a gene expression variant. It's not a coding sequence variant. So I think they are going to merge together. It's just that we're not quite there yet. This is all pretty new, this whole trying to map out regulatory variation and bring it into the predicting clinical phenotypes.
And thank you for a wonderful presentation. One quick question that might bring it back to one of the questions you asked the audience was, this is terribly complex. And it's actually more complex than I think most people are led to believe, that it's not just a matter of single SNPs. There's a huge regulatory component that's important in understanding what our DNA differences mean. Are we ready for consumer genomic information to be spread without the appropriate education that might be necessary in interpreting it?
Well, that's a loaded question.
Yeah. Well, no, and the idea is really to think about your thoughts and how should that play out?
Yeah, you'll hear a little bit of that at 5 o'clock. I'll put in the issue about genome to the masses as part of this. So my own belief is that we're at the cave person days right now. That is, we're incredibly naive about how to do this stuff, but there's still useful information to be garnered. If you think about it, I would argue family history, which we use all the time in our medical care, we incorporate that constantly. That's in worse shape than what we can get out of a genome, but most people don't agree with that, but I think that's true. I think your genome is a better record of your family history and your prediction than your family history is, which is not as good, but yet people are very comfortable with bringing family history. So I would argue that this information will be very valuable. We're not very good at it, but we can do something. And will we be better 5 years from now? Absolutely. Will we be better 10 years from now? Even better, yep. So I think it will be going forward. The issue about direct-to-consumer, I believe that you should be educating the people you're talking to. Most people, this group I hope does understand it pretty well, but the general population, they don't really understand statistics, or you wouldn't have so many people in Las Vegas probably. You have to educate people about probabilities and things, but I think most people can handle this if you tell them. And so I would argue this, but I would also argue at the end of the day, the person who's in charge of their genetic information is the person. It's not the doctor, it's not someone else. So I actually believe every person has a right to their DNA sequence and to learning this, but I think they should be told what it means and how to think about this. But the idea that it drives me nuts. Our IRB won't let us give our sequences back to the patients, which I find ludicrous because it's their sequence. It's not my sequence. And yes, it has certain errors, but I actually think we should be able to give it back. We just have to tell them, you know, the caveats and the things that go along with it. It's crazy that we can't do that. So that should leave you, if that got you riled up, come at 5 and you can clobber me some more. How's that?
Great, thanks. Didn't put you on the spot, I'm sure. It's
No, that's fine. Oh, yeah, it does. Yeah. It'll come. Yeah.
all sorts of recent things. So that was wonderful.
I think the 23andMe, what they didn't do is they didn't